It is a pleasure to introduce today's first speaker of the afternoon session, Dr. Mark Warren, Senior Director of Santa Clara Operations at BioIVT. Dr. Warren brings nearly 3 decades of experience in the drug transporter field and has played a key role in shaping our understanding of how transporters influence drug absorption, distribution, metabolism, and excretion, so obviously ADME. Today, he oversees all department functions at BioIVT's Santa Clara site, just down the road, and continues to support the scientific community through his deep knowledge of transporter science and ADME strategy. That is so important for drug development, for neurological diseases, and also how to deliver drugs to the CNS across the blood-brain barrier. Mark has been in the contract research sector for over 15 years, first at Optivia Biotechnology and since 2018 as part of BioIVT, following the acquisition of Optivia. In that time, he has partnered with clients to design and execute more than 1,000 transporter studies supporting IND filings. He is widely regarded for his expertise in interpreting how transporter mechanisms impact pharmacokinetics and drug-drug interactions, and how the interpretation of regulatory guidelines, which makes him a trusted resource and collaborator for both large pharma and biotech, and also for us internally at BioIVT. Before his work in contract research, Dr. Warren held research roles at Xenoport, where he helped advance several clinical candidates, including the FDA-approved drug for restless leg syndrome, by leveraging active transport mechanisms to improve pharmacokinetics. Mark also worked at Microsite Pharmaceuticals, where he focused on bacterial efflux transporters. And he holds a BS in genetics from UC Davis, an MS in chemistry, and a Ph.D. in chemistry and biochemistry from UC San Diego. And he completed his postdoctoral research at Penn State, where he studied enzyme reaction mechanisms. So with that, please join me in welcoming Dr. Mark Warren, who is going to present Transporting Your Way Around the CNS. Welcome, Mark.
Thank you, Tina. I get the unenviable task of being the first speaker after lunch. So after we've had a lot of protein, that tryptophan is being converted to serotonin, that serotonin is inhibiting those neurons and putting everybody to sleep. I get to be the excitatory one who manages to entertain and educate those neurons and bring you all back awake. So I'm going to talk today about transporting your way around the CNS. So a little bit of a background, a little bit of an outline of what I'm going to be talking about. I'll talk about transporters, some of the basics, some fun facts about transporters, tell you about the blood-brain barrier and transporters expressed at the blood-brain barrier, and how we might be able to use transporters to cross the blood-brain barrier. Then move into the central nervous system, tell you about the CNS transporters and drugs that are targeting those CNS transporters. And then I'm going to wrap up with something very new about Q and Q. And if you don't know what those are, you'll know by the end of the talk. And then finally a wrap up. So to make sure everybody's on the same page, because a lot of people here have a wide variety of backgrounds, the first question, what is a transporter anyway? Transporters, fast-paced, action, excitement, adventure. This is what transporters are on the macro level. When you drill down to what it looks like on a molecular level, you can see that transporters are membrane proteins that are responsible for moving molecules into and out of cells. And these molecules can be anything from drugs to nutrients, ion signaling molecules, waste products, or toxins. There's basically 2 types of transporters. Uptake transporters, which are responsible for moving molecules into cells, and efflux transporters that are responsible for moving those molecules out of cells. What kind of cells? Well, any type of cells, intestinal, kidney, liver, or brain. And this is really critical where there's any sort of barrier, like the intestine, where you have the barrier between the intestinal lumen and the bloodstream, or the kidney and liver, where you have a barrier between the bloodstream and either the urine or bile. But we'll focus more today on the brain. How many transporters are there? Well, a lot, hundreds that are expressed in the human body, that are grouped into 2 general categories. The SLC or solute carrier family of transporters. There are more than 400 of these, which are subdivided into 66 families. And typically, these are your uptake transporters. They're transporters like OAT1, or formerly known as SLC22A6. There's a second group are the APC transporters, the ATP binding cassette transporters. There's approximately 49 of these. People count anywhere between 48 and 51, depending on how you track. But these are in 7 families, and these are typically your efflux transporters. These are molecules like ABCB1, which is formerly known as P-glycoprotein, or PGP. Now, transporters, why are they important? Really because they control the ADME properties of drugs. They have clinically relevant effects on the pharmacokinetics and pharmacodynamics of drugs because they really control things, the absorption, like the intestinal transporters that are responsible for drug absorption, distribution, where it's going, metabolism, because, well, transporters aren't metabolic enzymes, but they deliver those parent drugs into the liver where they can be metabolized, and then they can transfer those, they can uptake or efflux those metabolites into the bile or bloodstream. And elimination. Transporters are really key elements in drug elimination, moving parent molecules and their metabolites into bile or urine. Now, the regulatory agencies are really focused on the absorption, metabolism, and elimination of drugs because that's where you get a lot of the drug-drug interactions. Not going to talk about that part today. What I'm going to be talking about more is the distribution, particularly how transporters are getting molecules into the brain or into the CNS. So after lunch, I got to give you a few fun facts because I've always got to add a few fun facts to my talks. This was a very early influence on my career. This was the first book I ever read, Green Eggs and Ham. I do not like green eggs and ham. I do not like them, Sam I am. By noted author, Dr. Seuss. Anybody not familiar with Dr. Seuss? He's a famous children's writer who has written a lot of books, very colorful images, pictures, cartoons, simple language that helps kids learn. And this is really what got me started on my path toward transporters. I'll come back to that because that's a complicated concept. I'll tell you about a couple of other things first, like the SLC45A2 transporter. This is one, if you've ever seen a white Bengal tiger, it's because of a polymorphism in the SLC45A2 gene, which causes the tigers to be white rather than their normal orange color. The SLC45A2, as National Geographic said, tiger, burning bright, just one gene turns it white. It's the same gene that's responsible for white coloration in horses. It's also one of 3 transporter genes that are determinants of skin coloration in humans. How about eggshell colors, since we're on that color theme? Well, it turns out eggshell colors are determined by pigments that are secreted when the eggs develop in the hen's uterus. Protoporphyrin, which is derived from blood hemoglobin, contributes to the brown eggshell coloration. In contrast, there's also blue eggs that have biliverdin or leucinin pigment in their shells. How do you get blue eggs? Well, that's the result of a transporter expression called OATP1B3. Normally, this transporter is only expressed in the liver, but because it has a retroviral insert upstream, the SLC01B3 gene, which encodes the OATP1B3 transporter, is expressed in the hen's uterus, and you end up getting blue eggs. It turns out, if you take chickens that lay blue eggs and crossbreed them with chickens that lay brown eggs, the result is chickens that lay green eggs. You see where this is going? I do so like green eggs and ham. Thank you, Sam I am. Because green eggs are due to transporters. And Dr. Seuss, back in 1960, wrote the first transporter book that I know of. Moving on to the blood-brain barrier. The blood-brain barrier, it's a barricade, it's a wall. Nothing ever gets through it, except maybe some things do. It's composed of endothelial cells, specialized endothelial cells around the blood capillaries, connected by tight junctions, proteins, claudin, jam proteins, and others, and a variety of other types of cells, glial cells, astrocytes, periocytes. It restricts the entry of nearly all molecules. But clearly, some molecules have to get into the brain. If the brain didn't get any glucose, it would not be a happy brain. So molecules like nutrients have to be able to get through this barrier. Waste molecules have to be able to get out. And really one of the key things that determines if something is getting into or out of the brain is transporters. Another thing I'll say, anybody that wants can have copies of my slides, and I have these clickable links at the bottom of these that reference papers, numerous papers along the way. At the blood-brain barrier, there's a lot of transporters expressed. I'm not going to go into this slide in any detail. I just want to show there's a lot of different transporters, uptake and efflux expressed at the blood-brain barrier, and I will talk about a number of these transporters a little bit later in my talk. Let's start out with some of the efflux transporters. In addition to all those physical barriers, the tight junctions, all the other cell types that are working to prevent molecules from coming in, sometimes molecules do get in, but we have efflux transporters expressed at the blood-brain barrier that work to prevent the uptake of drugs. They may get into those endothelial cells, they don't get into the brain itself because of molecules, because of transporters like PGP, P-glycoprotein, breast cancer-related or resistance protein, BCRP, and several MRPs, multi-drug resistance proteins, that are all well expressed at the blood-brain barrier and all act to prevent the uptake of drugs. Now, PGP is an incredibly efficient efflux transporter. Using our MDCK cells as a model to sort of mimic what's going on at the blood-brain barrier, we can look at how easy it is to get molecules, drugs like quinidine or digoxin across this barrier. If we apply them into the apical chamber and see how much can get through those cells into the basal chamber, with the basal chambers representing the brain, very little of them, because the molecules get in and PGP effluxes them right back out to the top. If we start with them, molecules in the basolateral chamber, they get through pretty easily. In fact, that difference between the transport from the B to A direction and the A to B direction, which is shown on this graph at the right, can be more than 100-fold. And so it's very difficult for these molecules to actually get through these cells and into the brain. Now, each of these efflux transporters can recognize many drugs, hundreds or even thousands of drugs, and very efficiently pump it out of cells back into the bloodstream. This is an example for PGP. Specificity? Not really. PGP is known to transport hundreds of different drugs, including everything on this slide. In essence, it transports the entire sigma catalog of compounds. And if you tried to look at these molecules and structurally align them, it's hard to find any structural elements other than say, well, they're drug-like molecules. Another point from this slide. Many drugs, their substrates are not just one transporter, efflux transporter, but multiple efflux transporter. So even if we inhibit one of these efflux transporters, the others can usually nearly compensate and completely prevent drug uptake. An example of this comes from lapatinib. Now, this is a tyrosine kinase inhibitor that's used in treating breast cancer. This is some work that was done by Joe Pauly and associates at GSK, where lapatinib was found to be a substrate of both PGP and BCRP. It has very, very low brain-to-plasma ratio. It's like 0.01 if you look at the amount that's actually in the brain relative to what's in the plasma. And this is due to both of these transporters. They took wild-type mice, and they knocked out BCRP. And what happens? You see a very, very slight increase. Okay, we'll take the wild-type mice, and we'll knock out PGP. What do you see? A little bit more. The permeability about doubles. So the brain-to-plasma ratio goes from 0.01 to 0.02. That's still not very high. But if you knock out both PGP and BCRP simultaneously, you have more than a 40-fold increase, 40 to 50-fold increase in the amount of lapatinib that's getting into the brain. And the two graphs at the right were sort of a low dose and high dose, which are showing the same effect. Which does lead to one idea. Can we improve drug uptake into the brain simply by inhibiting PGP? This really should work for drugs that are PGP substrates, but this has been done over and over. There was a first generation of PGP inhibitors like verapamil, which made it into the clinic, but only made it as far as phase 2 because of adverse events like hypotension, cardiotoxicity, and it really didn't show much clinical benefit. So a second generation of PGP inhibitors was tried, things like PSC 833 and others. Well, these made it phase 1, phase 2 again. You had cardiotoxicity, arrhythmia, and not enough activity in actually blocking PGP. So you had a third generation, molecules like alacridar. Again, these things made it into the clinic, phase 1, phase 2, even phase 3, but didn't show any clinical benefit. So the answer, can we really improve drug uptake into the brain by inhibiting PGP? The answer is probably not, because several of these PGP inhibitors have been developed and tested in clinical trials, and nothing has actually worked. They failed because of inhibitor toxicity or because of enhanced penetration accumulation of the drugs, not where you want them, but in non-CNS tissues. One of the challenges, PGP isn't just expressed at the blood-brain barrier, it's expressed everywhere. It's throughout the body. It's in the intestine, it's in the kidney, it's in the liver, and inhibition can lead to greater systemic exposure. So, in turn, this leads to dose-limiting toxicity or off-target effects of the drug you're trying to use. Another option, can we improve drug uptake into the brain by using Lipinski rule of 5 molecules? These are molecules that we've optimized the passive permeability, relatively small, less than 500 molecular weight. They're pretty lipophilic, C-log P values 1.5 to 2.5, small amount of polar surface area, not too many hydrogen bonds, uncharged. Well, that might work, but the problem is, again, these molecules are very non-selective. We can't control delivery into the brain. They're going to go everywhere in the body. So what can we do to get molecules into the brain? Well, one approach, let's try to design molecules that are not substrates of efflux transporters like PGP, or at least are weaker substrates. Can we look through and find some example where this has actually been done? Similar molecules that have different PGP activity. We can. If you look at opioids. Opioids, we have similar molecules with very different PGP activities. If we look at the structures of the two on the left, loperamide and fentanyl, these are very similar molecules. There's a lot of structural elements that are very much in common between these two drugs. Fentanyl, the poster child of the opioid epidemic and crisis. Loperamide, you can go down to Target and buy that. It's called Imodium. It's an anti-diarrhea medication. The effective dose of both of these is about 2 milligrams. Well, 2 milligrams is actually a fatal dose of fentanyl. 2 milligrams of loperamide treats your diarrhea just fine. No effect on the CNS. And really the primary reason why is because fentanyl is not a very good PGP substrate. It's a pretty weak PGP substrate. Loperamide is a phenomenal PGP substrate. And so as a result, loperamide has very, very low brain uptake versus fentanyl or morphine that have fairly high brain uptake, because PGP really limits not only loperamide absorption from the intestine, but also brain anything that does get through, it prevents the brain uptake. Loperamide is also metabolized very quickly by first-pass metabolism. But if we look at the oral bioavailability of these three opioid molecules, morphine, fentanyl, both of them have about 30% bioavailability. Loperamide, about 100-fold lower, like 0.3%. Very similar to what I showed at the early example with digoxin and quinidine, about 100-fold lower getting through. If we look at the brain uptake, and this was done by an in situ perfusion model, for morphine between wild-type mice and mice in which PGP had been knocked out, it's about a 1.24 fold difference, not really very much. Same thing for fentanyl, slightly higher in the knockout mice, but not very much. But for loperamide, we see a 10-fold higher brain exposure in the knockout mice than we do in the wild-type mice, indicating, okay, in these knockout mice, loperamide is accumulating in the brain, and in these mice, they're exhibiting opioid-mediated CNS effects from loperamide. So it is an opioid, it just can't get into the brain. So another approach, instead of trying to avoid PGP, because that's really difficult to do, what if we use uptake transporters to try to improve drug uptake into the brain? A lot of CNS drug targets, their receptors or transporters that are recognizing small polar molecules, molecules like serotonin, dopamine, norepinephrine, glutamate. These are not your Lipinski rule of 5 greaseball type compounds. They're very polar compounds. And there are a lot of SLC transporters that are expressed at the blood-brain barrier that can provide a way to transport drugs into the brain. And these include members of various families, organic anion transporter families, OATP and OAT, organic cation transporters, OCT or OCTN, nucleoside transporters, monocarboxylate transporters, and others, amino acid transporters like LAT1. I'm going to give you a few examples of these in the next few slides. The OATP transporters. In rats, there's OATP1A4. In humans, there's OATP1A2, and there's OATP2B1. But these are amongst the most highly expressed uptake transporters at the blood-brain barrier, and they're general transporters that have wide substrate recognition. OATP substrates include statins, prostaglandins, DHEAS, estradiol. These all exhibit pharmacological or physiological effects inside the brain, so these molecules are getting into the brain. For rosuvastatin, it's known that there are neuroprotective effects of statins because these have been observed in preclinical studies and in stroke patients that are given statins, who show a decrease in death rates, a lower risk of neurological deterioration, and a decrease in the mean infarction volume. Another example of molecules that are using the OATPs to get into the brain are triptans. Triptans like amitriptan, sumatriptan, they're used to treat migraine and cluster headaches. Clearly, they're getting into the brain. They're using OATP 1A2 to cross the blood-brain barrier. Of interesting note, triptans don't work in rodents. They don't cross the blood-brain barrier because they're not substrates of OATP 1A4. One of the challenges in this field, the OATP transporters are very different between species. So what may work in rodents or dogs may not work in humans because they're different things, or vice versa. But in rodents, there were no differences in brain accumulation of the triptans between wild type and OATP 1A4 knockout mice. But we know triptans work, they work in humans. So what if you take these knockout mice and you knock in human OATP 1A2 into these mice? If you do that, the zolmitriptan levels in the CNS were 1.6-fold higher than they were in the wild type controls, confirming that zolmitriptan is a substrate for OATP 1A2, but it's not a substrate of the rodent OATP 1A4. Another example, OATP 1A2, the uptake in this can overcome some PGP efflux. These were experiments done in our lab where we were looking at, on the graph at the right, the white bar looking at permeability from the apical to basolateral direction, again mimicking what you would see in the brain, going from the blood into the brain, of cells in which PGP and OATP1A2, there was no activity. So you see some permeability. In the middle, we add in PGP. And what it does, it drops more than 10-fold because PGP is not allowing this molecule. It's not allowing amitriptan into the brain. It becomes below the level of quantitation. But in these same cells, if we now add in OATP1A2, we see at least a 5-fold increase, showing that OATP1A2, even though this is a very good PGP substrate, which is normally excluding it from the brain, having it there can somewhat offset the effects of PGP and allowing drugs like triptans to cross the blood-brain barrier. Another class of transporters are the organic cation transporters, OCT1, OCT2, OCT3. This is another potential blood-brain barrier target. A drug like memantine, this uses the OCTs to cross the blood-brain barrier. Now, memantine functions as an antagonist of the NMDA, the N-methyl D-aspartate receptor, and it's used to attenuate excessive glutamate release in stroke victims. People that have strokes, the immediate effect is to decrease the oxygen and glucose levels, which causes an increase in calcium influx, increases glutamate release, leads to excitotoxicity, leads to neuronal injury and death. Memantine interferes at that calcium influx stage, and so it works to help as a protective effect in patients that have recently suffered a stroke, if you can catch them in time and give them this drug before too much damage is done. It's also been used to slow the progression of Alzheimer's disease and as a treatment for cognitive impairment or memory loss. Now, OAT2 and OAT3, organic anion transporters, these are also expressed at the blood-brain barrier. Dantrolene, an example here, it's used to treat spasticity after strokes, or it's been used in patients with cerebral palsy or multiple sclerosis. It also does appear to be neuroprotective, reducing excitotoxicity, apoptosis, neuroinflammation. Dantrolene, it's a substrate of both OAT2 and OAT3, which allows it to cross the blood-brain barrier and get into the brain. LAT1, this is a really interesting one because LAT1 mediates the transport of a lot of different drugs and prodrugs across the blood-brain barrier. Molecules like levodopa, gabapentin, baclofen, melphalan. What do these molecules have in common? Well, they're all LAT1 substrates, but they're used for a variety of different things. Everything from treating Parkinson's disease, epilepsy, for gabapentin, spasticity for baclofen, neuropathic pain, another thing for gabapentin, or cancer in the case of melphalan. LAT1 has very broad substrate specificity and high expression on both sides of the blood-brain barrier. It's on the luminal side and it's on the abluminal side, which makes it a very attractive target for brain-targeted drugs. This is an example taken from this paper that I have labeled here. These are a lot of different CNS drugs and derivatives that are designed to utilize LAT1. And if you go to this paper, there's actually 4 figures very much like this of various molecules that can utilize LAT1. There are a lot of additional transporters expressed at the blood-brain barrier. Excitatory amino acid transporters, EAAT, 1, 2, 3, ASCTs, serine, cysteine transporters. They transport small neutral amino acids. BGT, CAT1, CAT2, cationic transporters. SNAT1s, ENTs, which transport nucleosides. ENT4, which transports monoamine neurotransmitters. XCT and OCTNs. I'm not going to go into any detail on these, just giving you a list of some additional transporters that might be targets to use to get molecules across the blood-brain barrier. Summary for this part of the talk, there's a lot of uptake transporters at the blood-brain barrier that might be useful in getting drugs into the brain. And active transport can at least overcome efflux by PGP and other efflux transporters and allow passage into the brain, at least for some drugs. I want to switch gears a little bit and move from the blood-brain barrier into the central nervous system. In the central nervous system, there's a lot of neurotransmission transporters. Serotonin transporter, dopamine, norepinephrine, GABA transporters, glutamate transporters, glycine transporters, acetylcholine, others. I'll tell you a little bit about some of these. Serotonin transporter, a monoamine transporter that transports, of course, serotonin. And what it is, it's responsible for the serotonin reuptake from the synaptic cleft back into the presynaptic neuron. If there's dysregulation of the serotonin transporter, this impacts mood, emotional stability. And so there's a lot of drugs that are targeted at the serotonin receptor, or serotonin reuptake inhibitors, for example. If you inhibit SERT, serotonin transporter, this prolongs the serotonin signaling, which can be useful in some cases. This has been used in treatment for depression, anxiety, panic disorder, post-traumatic stress, OCD. And there's a lot of examples of these SSRIs, or serotonin reuptake transporters. Examples like Prozac and Zoloft and Paxil and Celexa. These are the ones you see the television ads for with people dancing through fields of flowers because these are your happy drugs, increasing your serotonin levels. There's also tricyclic antidepressants, which inhibit not only serotonin, but other transporters like norepinephrine. And these are used primarily for the treatment of depression, insomnia, migraine, OCD. And some examples of this are molecules like amitriptyline and others that I have on this slide. There's other drugs that inhibit serotonin transporter, like MDMA or ecstasy. This binds to certain and triggers a massive release of serotonin, leading to psychoactive and empathogenic effects. Cocaine is another example. It binds to serotonin transporter and the other monoamine transporters. Methamphetamine, that's another one that binds to serotonin transporter. That one's actually FDA approved for treatment of ADHD. How about the dopamine transporter? Well, the dopamine, that's another monoamine. And dopamine transporter is responsible for dopamine reuptake from the synaptic cleft, in this case back into the cytosol. Dopamine, it's essential for the reward and pleasure pathways. If you have disorders of DAT, it can lead to disorders, again, like ADHD, bipolar, depression, eating disorders, addiction, Parkinson's disease. And so drugs that target DAT, there are different categories here too. They can function as substrates, inhibitors, releasers, or allosteric modulators. One I want to point out in particular is probably with the most well-known, levodopa. This is a dopaminergic medication that's used to treat Parkinson's and some other conditions. Why is levodopa so special? Well, it's because of a dopamine deficiency is why you end up with things like Parkinson's. And it's a prodrug, which is capable of crossing the blood-brain barrier. It uses LAT1 to get in. Dopamine by itself can't cross that blood-brain barrier. You can give the patient all the dopamine you want, it's not going to get into the brain. Levodopa will. It's generally administered with carbidopa. Why? Because carbidopa will inhibit decarboxylase activity in the periphery and allow more of the levodopa to survive and cross into the blood-brain barrier. So what it does, it increases concentrations of dopamine in the synaptic cleft. There's also dopamine reuptake inhibitors, which again, you inhibit these, you can increase the extracellular dopamine concentrations and dopaminergic neurotransmission. These are also used for treatment of ADHD, narcolepsy, binge eating disorders, depression, smoking cessation. Anytime you have one of these dopamine, this has a high abuse potential because, again, these are feel-good drugs. Examples of this, bupropion or Wellbutrin, methylphenidate or dexmethylphenidate, and amphetamine. Other drugs that can target DAT, well, cocaine again, because that binds to DAT and blocks the reuptake, leading to a massive increase in extracellular dopamine, which is why it has a high abuse potential. Ketamine, phencyclidine, like PCPs, these are also dopamine reuptake inhibitors. And amphetamines, again, they cause efflux of dopamine into the synapse, leading to enhanced stimulation. There are medical uses, but they have a high abuse potential. Another neurotransmitter is norepinephrine. This is another monoamine transporter responsible for norepinephrine reuptake. Norepinephrine regulates attention and response actions, and dysregulation can lead to disorders like ADHD and psychiatric disorders. There are drugs that selectively transport norepinephrine transporter, or NRIs, like amitroxetine or Strattera, which is primarily used to treat ADHD again. It's very selective for NET. It doesn't hit the other monoamine transporters. So it increases the norepinephrine availability without affecting the other monoamines. Another one that's in development, reboxetine, this is again selective. This is used in the treatment of depression. It's approved in Europe, but it is not approved yet in the US. There are also drugs that are combination that inhibit both serotonin and norepinephrine. So these are dual reuptake inhibitors that block both of these transporters. Drugs like dualoxetine or Cymbalta, and these drugs are used for depression, anxiety, pain, fibromyalgia, depression, panic disorder, PTSD. There are also triple reuptake inhibitors, which block SERT, DAT, and NET. This might offer a more balanced amplification of the signaling, but there aren't a lot of examples of these. Amitifidine is under development right now for depression, alcoholism, and smoking cessation, but it's still under development. It hasn't been made it through yet. The things that we know block all of these, it is possible to block all of them. Cocaine and MDMA block all three of these neurotransmission transporters. The GABA transporters, GAT1, GAT2, GAT3, and BGT1, they help regulate inhibitory neurotransmission in the brain. They reabsorb GABA from the synaptic cleft. There's a couple of drugs that target the GABA transporters, like tagabine, which is an anticonvulsant used to treat epilepsy. Off-label, it's also been used for anxiety and panic disorder. And nipicotic acid. This is not used clinically because it just does not get into the brain, but it's used in research applications. If somebody wants to, they might be able to develop a prodrug of nipicotic acid that gets into the brain, and it could join this category of drugs that treat GABA transporters. Glutamate transporters, the excitatory amino acid transporters, and the vesicular glutamate transporters. Now, glutamate, it's the most abundant excitatory neurotransmitter in the CNS. So the EAATs, they remove glutamate from the synaptic cleft by reuptake into the glial cells and neurons. There's a couple of drugs that appear to target the glutamate receptors. I don't think anything has been designed to do this, but there are a couple that appear to do so. Ceftriaxone, an antibiotic, it's a beta-lactam which seems to upregulate EA2 expression, and it might have the potential to mitigate excitotoxic damage by reducing extracellular glutamate levels. And riluzole, which has been used as a treatment for ALS, which seems to stimulate glutamate clearance from the synapse. We also have glycine transporters, GlyT1, GlyT1, and GlyT2, because glycine is another inhibitory neurotransmitter in the central nervous system. The GlyTs mediate reuptake of glycine, again from the synaptic cleft back into the presynaptic neurons. A couple of drugs here, batopterin, which is a GlyT1 inhibitor. This was in clinical trials for treatment of schizophrenia, but it did not make it through. And a couple of compounds in development, which are selective GlyT2 inhibitors. These appear to be non-opioid analgesics that have potential use in neuropathic pain. In summary for the CNS, monoamine transporters like SERT, DAT, and NET, they're targets for a wide variety of neurological disorders, psychiatric disorders, and neurodegenerative diseases. The excitatory and inhibitory neurotransmission transporters like the EATs, GATs, GABAs, GLITs, these could also be drug targets for neurodegenerative diseases, epilepsy, and other indications. The real challenge, developing effective CNS medications. This requires solving two challenges simultaneously, two difficult challenges simultaneously. Optimizing your target specificity, finding something that's going to work on that particular target well, and having it still be able to cross the blood-brain barrier. Transporters are really the key to being able to do these things, so I'm always promoting that. It's the only way you can have the best medication possible, but if it doesn't get to its target, it's not going to work. So I want to conclude with something very new, Q and Q. What is Q? Well, this is queuosine and quinine. And queuosine is found in the wobble position of the anticodon of tRNAs, specific for asparagine, aspartic acid, histidine, and tyrosine. This is absolutely essential for eukaryotic life. But eukaryotes can't produce this. This is only produced by the bacteria, by the prokaryotic world. Queuine deficiency impairs aromatic amino acid hydroxylase enzymes, which leads to deficiencies in things like serotonin, dopamine, norepinephrine, and melatonin. Queuine deficiency might be one of the causes of diseases that are linked to these neurotransmitters. Now, queuosine and quinine, this is absolutely essential for brain function. We need this. We need to be able to synthesize proteins. It's essential for stress response and possibly for cancer prevention. I want to point out how new this actually is. The first article I have here is actually scheduled to be published in August this year. So that's how new this information actually is. How are these molecules getting into the brain? Well, this is also just recently found. There's a transporter called SLC35F2. This appears to be the sole transporter responsible for both queuine and queuosine uptake. Very highly specific, very high affinity, 67 and 174 nanomolar are the Km values for these. It's very selective for Q and Q. It's not a general transporter for other nucleosides or other nucleobases. And again, it's an EPUB at this point, which was just published last week. So this is the first time a transport mechanism is identified for queuosine and queuine. What do we know about SLC35F2? Well, we know deficiencies in queuine and queuosine are associated with various neurological issues due to deficiencies in serotonin, dopamine, norepinephrine. But what if you scan the literature, you'll find SLC35F2. There's only a handful of papers ever published about this, and they've described it as an oncogene. Overexpression of SLC45 or 35F2 has been correlated with a variety of cancers and with pretty poor prognosis in patients that have those cancers. So clearly, further characterizing and finding drugs to modulate the activity of the SLC35F2 could provide treatments for any number of diseases, anything from the neurological diseases involving serotonin, dopamine, norepinephrine, to diseases like cancer. This is one that appears you have to have the expression level right to be able to, for normal function. Being too far off in either direction can lead to disease states. So just in conclusion, I want to say transporters, they're key determinants how drugs can either gain entry into or be excluded from the brain. At least in some cases, using uptake transporters, you can overcome efflux transporters, which will allow your drugs to cross the blood-brain barrier and get into the brain. And CNS transporters, they're really attractive targets to treat any number of neurological and neurodegenerative diseases. Very brief commercial here. We have the world's largest collection of transporters available in my lab for a lot of the blood-brain barrier transporters, the CNS transporters. If you have any questions on any of these transporters, see me or the other guys from BioIVT. And another thing, if we don't already have the transporter assay available, our technology allows us to develop it pretty quickly. So if you're looking at animal models or anything and want to know more about these transporters, come talk to us. So with that, I will thank you and take any questions you have.